This colourful structure is a representation of an enzyme called amylase. Amylase is an enzyme that breaks down starches, which are long chains of sugars, down into simpler sugars. So if you were to put starchy biscuits into your mouth, like this person has done here, you would notice that amylase breaks down those longer chains of starches, and the biscuits would start to taste sweeter in your mouth. And hopefully you would actually recognize that this particular structure is actually a protein, and that is actually because all enzymes are proteins. So all enzymes are made of amino acids, and of course this is a quaternary enzyme. Each of the different colors in this enzyme, the green, the red, the yellow, the blue, and the light blue, represent a tertiary protein, and these tertiary proteins are bound together to form a quaternary protein. So enzymes are types of proteins, and as such they have a specific shape. And it is this shape that allows enzymes to bind with a specific reactant, which is also referred to as a substrate. So in this particular image here, this represents the substrate, and this is the enzyme. So for example, in the case of the decomposition of hydrogen peroxide, the enzyme would be catalase, and the substrate is the hydrogen peroxide. And enzymes catalyze reactions by coming into contact with the substrate at a very specific location called the active site. The active site is a specific shape that binds to one or more substrates. And when the substrate or substrates are bound to the active site of an enzyme, it forms something referred to as the enzyme substrate complex. And that's represented here by the substrate being bound to the enzyme. After the reaction is complete, the enzyme releases the products of the reaction. And this means the enzyme is ready to catalyze another reaction. Often, cells only need small quantities of enzymes because they are unchanged by the reaction and can be used again. There are two different models of how enzymes work: the lock and key model and the induced-fit model. The lock and key model suggests that the active site is highly specific for a particular substrate, so they fit together like a lock and key. And this is why most enzymes only catalyze one or a few chemical reactions. An enzyme that dismantles the fatty acid, for example, cannot break down the starch from an energy bar you've eaten, so it's specific to the reaction of dismantling fatty acids. So the lock and key model is actually represented by this image up here, where you can see that the active site is actually very specifically shaped to the substrate that fits it. In the induced-fit model, the active site of the enzyme does not fit the substrate precisely. In this model, the substrate molecule enters the active site and causes the enzyme molecule to change its shape until the two molecules fit together more closely. In this situation, the bonds within the substrate molecule are stretched as it interacts with the enzyme, and as a result of the stresses on the substrate, the activation energy required to start the reaction is lowered, and the products of the reaction are formed at a faster rate. So you can see, represented by this image here, that the active site is not going to fit the substrates perfectly. So as the substrates come into contact with the active site, they actually physically change the shape of the enzyme in order to fit before products are created. This particular illustration represents an anabolic reaction because you've started with two simpler molecules and ended up with one more complex molecule. So just remember, this could have easily been a representation of the opposite, a catabolic reaction. So enzymes can in fact, are in fact, involved in both catabolic and anabolic reactions. I mentioned activation energy earlier. Well, enzymes speed up the rate of reaction by lowering the activation energy, which is the initial amount of energy required to start a reaction. Even exergonic reactions, which release energy, require an initial bit of energy to get started. The enzyme brings the substrate into contact with it so that less energy is required for the reaction to proceed. So this graph represents how enzymes reduce activation energy, which is also often represented as E with a subscript A underneath it. So on the y-axis, you've got the potential energy of the molecules, and on the x-axis, you have the progress of the reaction, going from reactants to products. Now, without the enzyme, this much energy is required in order for that reaction to go from reactants to products. And you can see that by looking at how much energy is required in order for that reaction to go to completion. Whereas when you use an enzyme, you can see that you've actually reduced how much energy is required for that reaction to go to completion. So in fact, you're only using about this much energy. And this illustrates how you are actually using an enzyme to lower the activation energy required to start a reaction.